Hey, I'm Paul Hallam. I'm the Chief Operating Officer at Sims. My role is to oversee the facilities, the daily operation, coordinating the scientists and the facilities, maintaining the aquariums, basically keeping the place running for the research that goes on here.
My name's Shauna Murray and I'm a researcher in the School of Biotechnology and Biomolecular Sciences, and I'm in the process of moving to Sims to start research here. And our research is in a type of marine phytoplankton. So that's a microscopic organism that lives here in the harbour, everywhere in the marine environment, and produces a whole range of different types of toxins. Certain species do at various times. And our research is to identify the organisms and the toxins using molecular genetic tools and techniques. And that's what we're developing here at Sims.
So this is an ocean acidification facility. So as I was saying before, one of the major issues with climate change is not just the increase in temperature, but it's the fact that as CO2 in the atmosphere rises, CO2 levels in the ocean also rise, and that increases the acidity of the oceans. And in fact, if you had to pick one primary danger for the oceans from climate change, it's not temperature, it's ocean acidification. So for example, it dissolves calcium carbonate, which is one of the things that corals, snails, and many other organisms use to make their shells and their skeletons. So big risk, very big risk. And it's the main reason why people talk about the Great Barrier Reef disappearing in the next 30 or 40 years. And so what's happening here, there are tanks of compressed CO2 and it gets pumped into the water, and then you can manipulate exactly how much CO2 you pump into the water and how acidic that water then gets. And then you can put various organisms in those tanks and see how they respond.
My name's Kate Lee and I'm doing a master's project at Macquarie University looking into the movements of wobbegong sharks and eastern blue groper. So we're trying to assess their home ranges and how much area they need per an individual to look at how effective small marine protected areas are. My wobbegong work is down in Chowder Bay, Manly, and the blue groper work is in Bronte and Coogee, and we use acoustic telemetry. So we put these in the animals and they send off sound signals, and that picks up by our receivers, and through that we can use triangulation and determine exactly where the animals are 24 hours, 7 days a week.
I'm hungry. You know why fish are so smart? Because they swim in schools. Hey man, do you know where we are?
Chowder Bay.
What was that?
Chowder Bay.
I'm not following.
Chowder Bay.
Oh, chowder?
Chowder.
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 Okay.